So our final speaker today is Dr. Sorscher from Emory. Dr. Sorscher is a professor in the Department of Pediatrics, Division of Pulmonary Allergy, Immunology, Cystic Fibrosis, and Sleep at Emory. He earned his MD from Harvard. His research interests lie at the intersection of molecular pathogenesis and translational research, particularly concerning cancer and cystic fibrosis. Over the last 2 decades, he has advanced our understanding of cystic fibrosis and its treatment. Because of his contributions, he has earned induction into societies such as the American Society of Clinical Investigation and the Association of American Physicians. Today, he is going to talk about how precision therapeutics could be utilized for rare diseases such as cystic fibrosis. Welcome.
Thank you very much. It's a pleasure to be here. And I'm really glad to have an opportunity to speak. I want to thank everyone. This has been a phenomenal day of really formidable and informative and exciting presentations, which have been very enjoyable for me as someone outside the field. And I think part of the reason I've been invited to speak is because the work in cystic fibrosis is emblematic of a disease, a genetic disease, a really refractory genetic diseases like some of the hearing loss conditions we've been hearing about, Usher's and so forth, have been for so long. And that disease is not being adequately managed for many people. And so there are lessons learned there that might be applicable to the kinds of work that all of us do. And it's a pleasure to have a chance to present that today. So cystic fibrosis is not a disease of the auditory or vestibular system. It's sometimes called a generalized exocrinopathy. It's a disease that hits the secretory glands. So in the lungs, big secretions are made, they block the airways, they lead to respiratory failures. One patient described it to me once. It's like trying to breathe with someone's hands always over your face. The same thing in the pancreas and the liver, where big secretions block, they cause fibrosis and scarring, ultimately organ failure. It's interesting that in addition to these other exocrine glands, the sweat glands themselves are abnormal in cystic fibrosis. The sweat is very salty. The disease was actually discovered when in New York there was a heat wave and kids were coming in flaking skin off of their salt off of their skin. It's almost pathognomonic, meaning there are almost no other conditions that cause such salty sweat as in cystic fibrosis. When I was a boy, if you had cystic fibrosis, you would die when you were 6 or 7. When I was a medical student, maybe 20. And now people routinely live into their 50s and beyond. And that's because of medical research, like we've been hearing about all day today, these important conditions today as well. But in cystic fibrosis, there have been some recent, I think they can be characterized as breakthroughs that are worth mentioning, as they may have ramifications for other genetic diseases, like the ones we're interested in here. The disease is caused by a gene called the CFTR, or cystic fibrosis transmembrane conductance regulator. It's a 4,500 nucleotide, 4,500 amino acid protein with some functional domains. It sits in the membrane, in the plasma membrane, with membrane spanning alpha helices. If this is the membrane, this might be the airway, for example, the outside of the cell. This would be the inside of the cell. And 2 nucleotide binding domains come together, they heterodimerize, and when they do, this leads to a conformational change in the transmembrane regions, which opens a pore. And that pore allows the passage of chloride bicarbonate from the inside to the outside of the cell. There's also a regulatory domain that's not shown in this crystal structure because it's quite disordered and hard to capture in a functional way. But this is an ATP binding ion transporter dependent on phosphorylation in order for it to activate. We're going to next take a artist's rendition of what goes wrong in cystic fibrosis. This is a video I put together a few years ago, and what we're going to do is we're going to be bronchoscopically going into the back of the throat, and then into the airways, and then we'll come to the trachea, and then the bifurcation of the trachea, and we'll go into one main stem bronchus, and then we'll magnify. And when we magnify, we'll see the cells that line the airways with their cilia, rhythmically beating. We've heard a lot about cilia today. Over those cilia is a line of mucus, and we'll see that mucus being propelled, escalated to the back of the throat, where it can be coughed up, expectorated, or swallowed to destroy particulates or bacteria that land in the lung. And then we'll magnify again, and we'll go between 2 cilia in this artist's rendition. We'll see the CFTR molecules, these channels that transport chloride bicarbonate. We'll see the chloride bicarbonate represented as little yellow balls, and they will be passing from the inside to the outside into the airways. When those balls move, water follows passively in part through aquaporin channels. And that allows the cilia to normally extend. When it extends, it can beat and it can move mucus. And then we'll come out again and we'll ask, what would happen if that process was disrupted. If suddenly those channels weren't working so that none of those balls, none of those ions could move, none of the water would move, the cilia would then not have a normal airway surface liquid. The periciliary depth would be depleted. The cilia would bend over and hunch down, and the mucus would build up and basically compress them on the surface, plaster them to the surface of the airways. In that case, mucus continues to accumulate, becomes infected, becomes inflamed, and then you die. You die because you have mucus obstructing your airways and you can't clear it. So let's go and see what this looks like then in this hardest rendition. Here we go. So over the tongue and through the vocal cords. And now we're going into the trachea and the bifurcation and into a main stem bronchus. And now we're going to magnify. And now here, the epithelial cells lining the airways. This is the airspace out here. You can see the cilia beating and moving the mucus up to the back of the throat. So this is the mucociliary escalator. And now we're going to go between two cilia and look at the CFTR, which is represented as these guys, the inside of the cell, the outside of the cell, and chloride and bicarbonate passing out. When the phosphorylation happens, the channel opens and it gates. We come out again, we see the cilia still beating. And now we're going to ask, what would happen if that mechanism became defective? No ion movement, no water movement. That's depicted here. Where now you see channels that are locked shut. None of the chloride and bicarbonate can move, nothing gets out, no water moves. And as a result, we come back out again. At a molecular level, the cilia can't extend properly, the fluid's depleted, the depth decreases, the plastering of the cilia to the surface, the accumulation of mucus, and then in this artist's depiction, bacterial infection and inflammation, which is a good model, the best, maybe the prevalent model for a good part of the reason that people with cystic fibrosis get sick and die. So what kinds of things can go wrong with the CFTR? And it's a lot like what we've heard about today, and the answer is a lot. There are more than 2,000 different mutations that have been implicated as causing a disruption of this mechanism. And when there are 2,000 mutations, as we've heard about today, that presents a problem for personalized or precision type interventions. Lots of things can go wrong. They might not be doing it all in the same way. So a therapy that's tailored to a specific kind of problem maybe needs to think about the specific problem it's being used to address. And in CF, what we find is that although there are 2,000 mutations, they can be grouped into about 5 groups. And when Obama got up many years ago and announced the Precision Medicine Initiative, he actually had a patient in the audience with CF stand up as sort of the poster child, literally, of this disease and how precision or personalized medicine might be useful. You can divide the CFTR mutations into 5 categories. There are premature stop codons that interrupt protein synthesis. There are problems with maturational processing where the protein is made, but it's missing something and it's misfolded. The normal cellular quality control mechanisms that detect and degrade misfolded proteins sends it to a trash bin, to the proteasome, or to another degradative pathway so that it can never make it to the cell surface to do its job. There are mutations that basically lead to locking shut of that channel, like in that cartoon I showed a minute ago. No balls moving, no ions moving, and the cilia fall apart. So this is no CFTR at the surface because it's been degraded. This is CFTR at the surface, but just locked shut. There are problems with stability at the cell surface and its recycling and degradation, and then also just splicing defects. This is the common mutation. In about 70% of cases, it's a phenylalanine deletion at position 508 of this 4,500 amino acid protein. So there are some mutations that are very common. I think 90% of people have an F508del, many homozygotes. There are some that are less common but have a different mechanism. And so the concept here is that if you're thinking about precision interventions, you can develop a drug that might be really good for reprocessing this, retrafficking it, and getting it to the cell surface. So that drug might have no value at all against the gate that's there and already locked shut. You could tailor a therapy, a precise therapy, to open a gate that's locked shut, but it would do nothing if the protein's not getting to the cell surface. So beyond understanding the genetic defects, what's been learned in our field is that it's really important to understand the protein abnormality and the molecular mechanisms so that the therapies can be well tailored. That's, of course, outside of the domain of genetic interventions. If you're doing a nucleotide-based therapy with an mRNA or with an editing strategy, and if it's done right, it's kind of agnostic to what the problem is. You're going to fix the whole protein at the same time. In the case of what I'm going to present today, which is mostly small molecule type interventions and an approach that could be used in many cases of hearing loss, it's different in that we use small molecules that are given orally instead of genetic interventions. But I think that's a consideration for diseases of hearing and the auditory system. I think small molecule interventions, and we've heard about some of them today, could be valuable And I'm going to offer that this approach in CF has worked in many of these different kinds of things that can go wrong. So there are drugs that cause read-through of stops, trafficking of these sorts of proteins, small molecules that can address surface stability, or gates that are locked shut. If you can design the assays, I'll describe in a minute, there's the potential for finding a small molecule that can be taken orally and used as an intervention, or given reading. Okay, so this is that approach. Probably everyone here is familiar with it. It's just high throughput compound library screening. And for those who don't know, it works like this. You give a robot 1,000,000 drugs, and then you take a dish that has 1,000 wells, and you put in that dish cells that have a defective CFTR with one of those defects, a specific defect, a defect that we understand the mechanism of. This has kind of turned traditional pharmaceutical sciences on its head, because the decision was consciously made in the CF world to say, okay, we know it's an ion channel, we know it moves ions, that's all we need to know. We know we can engineer a cell so that if it's working, the cell is very bright. And if it's not working, the cell is very dim. And so you've got a dish with 1,000 wells of cells like that, they're all dim. And then you add drug number 1, the robot does to well number 1, and drug number 2 to well number 2 until 1,000 drugs have been added, and then that's put on the shelf. The robot then takes the next dish, does the next 1,000 drugs, the next thing, and comes back 2 days later and looks at 1,000,000 wells, or 2 weeks later, looks at 1,000,000 wells and says, wait a minute, these 6 are very bright. So those drugs become candidates for fixing a specific kind of problem in the CFTR, and that problem, those drugs then get moved forward through the traditional path, through medicinal chemistry, through structure activity and SAR, ultimately in animal studies. And what's been learned is every time this has been tried in CF for a number of different kinds of mutations, there's been a good result. And so it's worth considering. In our lab now, we have projects not just in cystic fibrosis, but we have a project directed towards Pompe, towards adrenal leukodystrophy, and towards sickle cell, all of which the kind of approach. Screen a lot of drugs and have serendipity work for you in a way that can be very valuable. Some of this has been done before, of course, in this field, but it's just that CF is a good example of how it can be useful, and how it might work. Okay, so the kinds of drugs that have been identified that have now been clinically approved and that are helping people with this disease to live longer and better lives are generally categorized into 2 groups: the correctors, which take the common class 2 mutations that are misprocessed, they're sent for degradation, they never make it to the cell surface. It binds to a misfolded CFTR, it corrects the folding so that the cell leaves it alone, does not route it to degradation, gets to the cell surface, it does its job. And the potentiators, which take a gate that's locked shut, like the one I showed you in that video, nothing's getting through, a small molecule moves in, binds to CFTR, changes its conformation, and makes it capable then of transporting anions. This is just to show a picture of a short video of how this works. Here's the CFTR, the outside of the cell, like the airways, the inside. You'll see at first, none of these yellow balls are moving, so no water's going, and the cilia can't extend. And then you'll see a drug come in and glom on, bind the CFTR, change its conformation. And there are crystal structures now of the binding sites for these drugs that have been identified through high throughput screening, and you'll see how that works. Here's just a short video to depict that. Drug binds, protein changes, and the ions. So that's just a visual of something that's pretty fundamental to the work that's been done in CF. And the point to make again is that drug wasn't identified with any sort of real knowledge base, either about the crystal structure of the protein or about what might or might not bind, or with any kind of in silico modeling. Hundreds of thousands of drugs screened. There's one that opens the channel. Then later we go back and find out what exactly is it doing to the conformation of that channel. Yes, I do think, though, it underscores the importance of understanding the mechanism that you're dealing with. You know it's a mutation, you know it's a missense, you know it's probably causing a folding abnormality. What does that do functionally? We've seen many great presentations today about understanding function in this kind of setting. So what do these drugs do for people? This is a very sort of standard response, usually within a week. Often within a week, people are having kind of a life-changing experience. It's been predicted by some people that these drugs may add decades to the lives of people are dying. This woman, I remember the day before I took my first 2 pills, constant coughing for decades, infections, antibiotics, anxiety, and depression, and the knowledge that CF would one day take my life. The after has been complete disappearance of my cough, stop antibiotics as my lungs are clear, feeling optimistic that CF might not take me out after all. So it's really been an important thing sort of to watch these kinds of developments. This is another example. I knew to expect what's known as the purge. Patients talk about this. They take the pills, and within a couple of days, they're having all of this mucus that's been there for a long time come up, and they can breathe much better. It's like a dam that just broke. By the next morning, there was no junk coming up, no wheeze, and no. So it's really been remarkable to watch this happen, and it came in some ways, a very non-scientific way. It came by screening enough compounds so that we found something that worked, and having the right endpoint so that you can make a decision about whether you're on the right track. This is the work of many, many laboratories collaborating. And one of the beauties of a day like this, which is very evident to an outsider, is there are many laboratories and they work really well together. So there's the possibility to come together. I've already had 2 conversations today with people who said, yeah, there's a common defect that causes hearing loss. It might be worth it to take a look at that in this sense context. And so I think it's a wonderful meeting. A couple of points about the modulators. They're approved for about 90% of the FDA is approved. But there are 10% that are unlikely to get approval because they have mutations that are really hard to address with a small molecule. They might be bad nonsense defects or splice defects or very bad folding abnormalities with big indels that you can't really get at with a small molecule. And what that means is that while for 30 or 40 or 50 years, everyone with CF has been in the same boat. Little kids all know their friends who have CF and they support each other. Suddenly now 90% are on one very good boat and 10% are on a very different boat. And so it makes it almost a mandate to figure out a way to get to the last 10%, as they're often called. Those are at risk of being left behind by the field because they don't have a drug. And that's what sort of talked about on a day-to-day basis. And I want to just mention that it's not all rosy, that there are some really significant challenges that remain. One of the challenges is that there are 2,000 mutations that cause disease, and some of them are really rare or even private. You might be the only one in the world with a certain form of cystic fibrosis, and if you're the only one in the world, it's really hard to get your mutation approved by the FDA to get on label for the drug, because the FDA approved drugs, right, based on double-blind, placebo-controlled trials that show something that's a real benefit. And if you're the only one in the world or one of 5, it's been estimated there are 1,000 mutations, which are less than 5 patients each. And if that's the case, there isn't a control group and there's no way to do that study. So that's been a major problem in terms of getting to the last 10%. Another problem, we've heard a lot about this today in the space that this group in this field looks at, is the complexity of the molecular phenotype for specific variants. There are 5 categories, and it's not as simple as you're class 2, so you have this problem, and you're class 1, so you have this problem. There's overlap. And so you might tailor a therapy for class 3 and find it also works in class 4, for example. And that's, I think, one of the important lessons learned in our setting. Another is genetic heterogeneity, something else we've heard a lot about today. And that is that there are people who have the same genotype, siblings who have the same genotype. So they have the same 2 mutations in this autosomal recessive disease, and they have very different clinical course. Or you give them both the same medicine and they have very different outcomes. And this has been looked at hard to see what are we missing. The twin studies have been done. It's not and the modifier studies have been done, the GWAS studies, and it's very hard to get a handle on what this is. There's a lot of missing heritability in this field. It's not known what it's due to. A lot are interested in epigenetic changes that might be important. And I think one thing that's come out is that you may have the mutation that causes disease in CFTR, but you may have 50 other polymorphisms in your CFTR that might in small ways be contributing to the overall ability of the protein to do its job. And so focus has shifted in some ways in our field very recently from let's look genome-wide to let's take a really good look again at CFTR. It's about 180,000 base pairs if you count the non-coding stuff, just 4,500 coding. So there's a lot of information in there that controls things like mRNA levels, regulation, everything else that could be important that we might not have accounted for and won't show up on GWAS often. So that's another lesson I think that we've learned and we'll continue to track. This is the same as mRNA variability. And then I want to spend a minute talking about this elephant in the room. And the cost of these drugs is what the market will bear. And I think everyone's aware that for all of these leading edge therapies, like the ones that are coming online, I'm sure for hearing related issues as well, for the sickle cell problem, there was an article in the New York Times last week, $3 million to have your sickle cell fixed, and that will not have any market share in Africa and Asia and India and other places where there's a lot of sickle cell. And even in the United States, it'll be a challenge. The cost of these cystic fibrosis drugs, which are life altering and add potentially decades to life, it's $300,000 a year for the rest of your life. So you can't afford it, so most. In the United States, if your insurance company will pay for it, you can get it. And most insurance companies will be, in part, not to take anything away from either the company Vertex, which is charging this much for the drug because they've done a miraculous thing. Wouldn't want to take that away from the patients or for them, or for the insurers who are faced with a difficult situation where how can they say no? There are kids that are going to die of this disease and they're going to hold it back. It's worth the $300,000. And you can make arguments that it keeps people out of the hospital, gives them a normal life, saves in other ways. But it is an issue. Cost is an issue. Now, it's worth mentioning that most of the drugs that are prescribed in the United States by people like me, they're given off label. If there's an antibiotic for a certain indication, and I want to give it for another indication because I think it'll help, I write the prescription, I give it to the patient, they go and they get the antibiotic and they take it. It doesn't have to be FDA approved for that use. It just has to be on label for something, and I can write the prescription. So if there were a patient living in, I don't know, rural Montana, who had a really rare form of CF, and I wanted to say, hey, is this drug going to help you, which helps everybody else, and they weren't on label, I could write them the prescription, I could give it to them, and they could get the drug if they could pay for it, but they would take it to the pharmacy, and they would say, okay, give me $300,000, and that would be the end of that attempt. So the problem is not just scientific, the problem is social, economic, cultural, that we have really expensive drugs that will help people, but we can't get them. In this country, we get them to 90% of people. The insurance companies will pay for them. In other places, not so much. There are lots of places where you can't get the drug simply because they're so expensive. That'll be important. It's something I'll say more about. These are the genotypes among the 2,000 that have been approved for modulators. It's a large list. Some of them are very rare. This is not to memorize. This is just to give you a feel. And this is one of the things that's been done about the problem with that patient in Montana. There's no way that patient who might have a unique mutation can ever have a double-blind placebo-controlled trial. That's the only person in the world who has that form of the disease. So after a lot of lobbying and a lot of data, the FDA has made sort of a concession that will be relevant, I think, to similar approaches in this room, whether they're genetic or small molecule, whatever they are, with regard to hearing loss. So the concession is, if you have a really good in vitro system that can really predict who might or might not respond to a drug, and you can show that for that really rare variant, that the drug works in that system and it's been well linked to the clinical outcome, in the same way that if your cholesterol falls, they'll prove a drug. You can't tell your cholesterol's fall, but it will help you with your heart and with the risk of stroke. If you can have a surrogate, an in vitro surrogate that works, the FDA has now set a precedent in cystic fibrosis that allows you to move the drug forward. Does that make sense? What I'm saying? So this is just an example of some of the data. This is what happens with sweat chloride as you go from having CF to something that's kind of like CF, really probably not CF to definitely not CF. You can see that the chloride is falling in the sweat. That's a really good in vivo surrogate. And this is what happens in a cell model called the FRT line and in other in vitro lines when you ask about the same mutations that are in these 80 or 85, 86 patients. You can see that as you go from having CF, there's a threshold of about 10%. Not really CF, to definitely not CF, but something's the matter to nothing's wrong. You can see those numbers go up. And so if you have that patient's genotype and you take that patient's DNA, you put it into this cell model, you ask, how well does it work as an anion channel? And you see, it works really well. It works like this. The FDA will approve it under the right conditions if Vertex sends it as part of that. So that's one solution that's been worked out for cystic fibrosis that I think has implications in many contexts, including here. And this is just to make the point I made a second ago. Each of these horizontal lines is a CFTR, and each of the vertical ticks is a polymorphism that doesn't cause disease, but is on the background haplotype of a CFTR mutation. And you can see there's a lot of variability here. And the question is, when we take two patients that have the same genotype and they behave very differently, is some of that because they might have very different background polymorphisms on those two haplotypes. They might be doing things at the message level, protein folding, the ability to gate and so forth. And that's another lesson that we've learned, maybe the hard way. But we have to look closely at CFTR in addition to everywhere else in the genome and maybe epigenetics as well. We just published last year, this is relevant in this context, when you have a gene that's 180,000 nucleotides long, and you have ticks all along it of polymorphisms that you don't think cause disease, and you sequence, what you get is you get all the polymorphisms, but you don't know who goes with whom, unless you're doing sequencing sort of long reads and you're properly phasing. You need to know what's in cis and what's in trans. And now new techniques like the high C technique or like just long reads are making it possible to do that. And I think there's intense interest in our field, for example. Just in the last couple of years, 5,000 CFTR genomes have been sequenced, 5,000 CFTR genomes project, it's very originally called. And those 5,000 genomes, there's strong interest in what are the other polymorphisms that might contribute to phenotype and who goes with whom in terms of cis and trans. I'm going to close just by talking about one other approach to this problem of rare variants. Our lab does a lot of work with rare variants, and the last 10%, that's sort of become our passion. Try to figure a way that people that can't get these drugs can either get them or we can make other drugs for them that might work. So that's what our laboratory works on mostly now. And this is a mutation called N13L3K. It's a relatively common mutation. It's the second most common class 2 defect. A lot of people around the world have it. It's not on label. So if you have this mutation and you want to get the drug, you have to pay for it or you can't get it. And Vertex has been very reluctant to help people with any kind of humanitarian or special permission to use the drugs. They don't give it away on a compassionate basis very much. So they have a lot of interest in this study because at $300,000 a year for 1,500 patients, for example, that's a lot of money that they could be making if this would be approved. When they put this mutation in their FRT model, though, the model that I showed you a minute ago, the in vitro model that the FDA will use for approvals, it didn't make the 10% threshold, and so they didn't go for it. But we got a bunch of labs together and screened in a lot of different ways to ask, well, how does this mutation do in other systems? And this is the data. I won't spend a lot of time going through this is just a bioelectric measurement of how much ion channel activity there is. An upward deflection means, the upward deflection in red means for N13L3K, we're getting activity. And the activity is 26% of wild type, 25% of wild type, and it's well above the 10% threshold. So we went to the NIH and to the CF Foundation, said we'd like to do a trial of N13L3K patients. We believe the effect size will be large. We think if we give them the drug for a month, we'll have people who are having purges and bringing up the mucus. And there was a lot of understandable skepticism because first of all, we didn't have a control group. And secondly, because it's got to be a large effect size. There aren't that many patients. And so we were proposing 20 patients just to see what would happen, just to see if we would be right, that our in vitro data using the well-established model systems could make a difference. And this study's getting a lot of interest all over the world. We've gotten letters from Russia, from South America, from Asia, from South Africa, patients who wanted to come here to take part in the study. It was limited to the United States because it was more than just a study of N13L3K. It's a study of whether we have really good scientific rationale and really good in vitro data. Can you make some predictions that might benefit people? And so we brought them in, and we brought them in just for 28 days of treatment. You can often see effects after a few days. And we followed this with a washout period where we stopped the drug to see if whatever effects we saw would go away. And we looked at endpoints like their sweat chloride and their lung function, which is a great deal of interest for label expansion, and their weight, and a quality of life questionnaire. And briefly, this is just the data. Everybody's FEV1 except one patient. Everyone's lung function improved. People had a significant improvement in quality of life. It is that's an endpoint that can have a fair amount of ascertainment bias. You're on a drug to help you. How do you feel today? You often feel better when you're on the drug, but this is not as effort dependent. This is, and they also had an increase in weight. And now Vertex is using this data to go to the FDA and see what kind of ground they can gain with this clinical data. If they can't, then they'll run a bigger trial because it's worth it. But what this indicates is the ability to do a study like this and to make the case that we might be missing people. So now we're looking at larger groups of patients who we think based on our in vitro findings could be useful, that the drug could be helpful in them, and we're running those kinds of studies as well. This is just to say that in our little study of 20 patients, 16 of them took their data to the insurers and they said, I got better and I need this drug. And 80% of them got approved on the drug where they hadn't gotten it before. So if nothing else, it's a very satisfying study for that reason. And then This is just to say when you've got an ultra rare variant, these are where the patients, some of them came from for the study. We flew them in 4 or 5 times for the study, gave them the drug, and certainly endpoint went well. So to summarize what I've said, even with an incomplete knowledge regarding a genetic disease, compound library screening can repeatedly identify small molecules that can address certain molecular abnormalities. You hit them with so many drugs, you find some that bind and do what you hope they'll do. And that's been repeated, you see it many times. The modulators have really transformed care and prognosis. And transformative is a word that I think is not used lightly, but it's really what's happening in this field. They've only been around, some of them, for 3 years. And whether in 10 years or 20 years or 30 years, they'll still be doing this, or whether there'll be side effects we can't anticipate or tachyphylaxis we don't know. So far, things look good. And I've described a few challenges to precision medicine, which I think have relevance across the board. Gaining regulatory approval for ultra rare indications is tough, and it can be tough, and it has to be approached with the agency very thoroughly. The disease heterogeneity and a measure of phenotypic imprecision have to be considered. There can be a lot of differences, and we've heard a lot about those in the hearing space today as well, and then high drug costs. And then just to finish, there's intense interest now for the people in the last 10% are doing this genetically. It's not as easy as it is in the eye or in the liver, or maybe ex vivo in the blood. How easy it'll be in the ear. It's really exciting. When they show that picture of that kid from the New York Times today, how can you not really be inspired? I mean, it's inspiring. And there's real hope, and that's a great thing. And that's much less precise. That can be done with, got the gene, make a super exon, you put it in with editing, and then you fix everybody. It's a great, it's a great opportunity. It's very inspiring. This is a project in our lab looking at the nanoparticles for delivery to lung, where it's not as easy. You blow things down the lung. The original thought was, oh, great, we have all these. First, we have easy access, right? Second, we have all these viruses that are really good at getting in the lung. And third, we can put the CFTR in there. So let's just do it. And what we find, this is not many years ago, is that the lung is also very good at keeping those things out. And if you got a lung that's already sick, you get infection and inflammation, and you have things that you're adding. It's not easy. So the delivery aspects of CF are a major challenge that might not be present in the ear, for example, and really makes one hopeful and excited about that. And I'll just conclude by thanking people at Emory at University of Birmingham, Texas. Glad to take any questions. And just to say again, it's really been a great day for me. Thank you for having me so much. You.
Yes, I'd like to echo what you said about it being an absolutely great day from all the speakers, and this has been a wonderful presentation for the last talk of the day. So thank you. I believe it was 3 or 4 slides before this where you showed where they were giving the drugs off label, and you had a huge variety in how some patients were doing well and others weren't doing well, and then one did not do well at all. Do you think this variation in the response of those individuals, do they represent where there may be more than one variant in a particular gene, or variants in more than one gene that is causing that variation in the response?
So I'll repeat the question just in case you didn't hear it. The question is, there are patients who get these drugs in the 90%, and about 5% of them don't do well with drugs. It just doesn't touch it. And that seems to be genotype independent. It's not, they can have the same genotype as everybody else. So it's not just, you look at F508del is the common mutation. You look at homozygous individuals, and you can see quite a discrepancy in about 5% they don't do well. Most people do very well, some don't. And the question is, why is that? And the answer is no one knows. And it might be they have many polymorphisms in their CFTR. It might be it's not known yet that they have modifiers elsewhere in the genome that are contributing to this. There's a lot of interest in the pharmacokinetics of the drug. Maybe the drug is just metabolized more rapidly. That's been looked at. It doesn't seem to be the case, but those are the kinds of ideas. There are also questions about tissue penetrance. Might be important in different people. So all kinds of ideas come up when you see something like this. And in a way, it's not surprising. Every drug has quite a bit of variability in its response. But we're in a world where we understand what's gone wrong here in a way that many other diseases don't have that fortune. So we hope we'll be able to learn more specific things about that. Really good question. You know, excellent question.
Thank you.
Any other questions?
What is the downstream of CFTR apart from exchange of these liquid fuels?
Yeah. The question is sort of, well, does CFTR do anything else other than what you showed us? And I think the answer, it's a great question. And there's a lot of literature about other things that it might do or modulate or mediate. There's interest, for example, in what it might do to inflammation or to susceptibility to infection or to tissue remodeling. And there is, for each of these, a school of thought that argues that they have data that there might be a contribution to pathogenesis from these other pathways. I think the generic or the general answer would be all are very interesting, but none have really borne out the way the idea that we have a problem with fluid and electrolyte secretion, and we have a problem with mucus that's thick that happens in the pancreas and the lung and the reproductive tract and in the liver and other tissues. So I think, from the standpoint of devising therapies, those are really important to understand better, but they're not sort of in the critical path to therapeutics. Another way to say the same thing is, I think if you looked, like we saw earlier today in a presentation at the transcriptome of people with CF and who don't have CF, you would see a lot of changes other than CFTR, because there are a lot of things that just the ion tone in the cell might regulate. So you'd learn a lot, but I don't think that's not been the sort of the main thrust of the work. So good.
Just a follow-up one. So it's a very especially CFTR. I mean, CF is a chronic disease.
Lifelong.
And then if it is the case, so what your drug is doing is definitely doing a wonderful job of opening the channels and exchanging. But that kind of mechanical stuff is doing. But if it is a chronic, are you considering any other inflammatory and bacteria? Because bacteria plays a very important role.
Yes, it's a great question. I didn't mean to imply at all that that when you take this drug, your CF is gone and you never have any problems anymore. If you've had 20 years of damage in your lung, you're not done, and you'll still take antibiotics. You'll still need anti-inflammatory molecules potentially, and you'll still need drugs to treat some of the pancreatic dysfunction or the hepatic dysfunction. So it isn't a problem solved. It's just a problem much, much better, exactly as you're saying. And I think that's the best answer to that question, that we still have to continue to work hard, and that we don't, there's a feeling that if you start these drugs in kids or they have a lot of lung damage, that you'll be able to prevent them from ever getting sick, but it would be premature to say that.
Quick question, that you receive patient you treated with topramycin, do you find any patient with aminoglycoside toxicity?
Very common. The question is about aminoglycoside toxicity in people with CF. And they get gentamicin and they get Toby and they get other drugs and they get hearing loss. And it's an area of intense interest. And I'm really glad you asked the question, because the Cystic Fibrosis Foundation, who funds a lot of research in this area, they have a $3.5 billion, $4 billion budget because they have made money. They invested. It's a really interesting story. They didn't have a lot of money, but then they invested in a company that did this high-throughput screen, the first company to develop these drugs. And then those drugs, when the company, they had nothing to do with the charge for the drugs, but when the company started charging $300,000 a year for the drug, they got royalties. They got so much in the way of royalties that people started coming to them and saying, whoa, I can't afford the drug, and you're the foundation and not for profit, what's going on? So they sold all their royalty rights after they got $3.5 billion. So they're really interested in this problem of aminoglycoside toxicity. And whether people with CF, because of a problem with another transporter, are somehow predisposed, I think that's an area of some interest. And I also mentioned that people in the last 10%, some of them, they have premature stop codons in their CFTR. When you have a stop codon, one of the strategies that's evolved is to read through the stop codon. In other words, poison the ribosome just enough so that it takes a non-cognate tRNA and it sticks it in there in place, and then you get a full-length protein that might be working. And that's a holy grail, because if it worked in CF, it would work in every other context. If you had a really good read-through agent, that would be a really great thing for many genetic diseases, or most genetic diseases have stop codons, including hearing loss, of course. And so there's been a lot of interest in in drugs like aminoglycosides, because the prototype is a drug called G418, which is a aminoglycoside antibiotic. You're familiar with it, right? It's a commonly used drug, and it causes read-through, CFTR. You can get some full-length protein. We published a paper on that 20 years ago now. A company picked it up and made analogs for aminoglycosides, but nothing has either surmounted the problem of toxicity or been effective enough that I don't think we can get to the 10% level yet. There's a lot of work in that area. So that's another place where there would be a lot of
I
open.
still use what is the drug is aminoglycoside, still the topramycin or.
Tobramycin.
So as we did the topramycin drug sensitivity to the, I presented in the morning, they have some dosage issues to if we have the mitochondrial mutation.
Right.
And I was at Cincinnati Children's, we screening these mutations.
Yeah. Cincinnati Children's is a powerhouse in my field. Yeah. Yes.
More of a comment. I think your lab's focus on rare mutations that affect very few and trying to solve that is absolutely extraordinary and commendable. It's remarkable.
You're kind, and I really think it's so. I mean, if you knew when you see people that are in that last group, it's really it's compelling. You know, we had a conversation about this yesterday where there's fundraising for CF, and when you've got 90% helped and 10% not helped, it's like you have a new genetic disease where there are people that aren't being helped, and that's how we feel like we're dealing with a new disease now. Thank you for that kind comments. Yes.
So as with the ion channel opening this drug, I have two questions. One is, what's the half-life of this drug? And secondly, do you anticipate being an ion channel opener in oversensitization?
I mean, the first one is the half-life of the drug that opens the channel. So it's a twice-a-day drug. So its half-life is, a day. But Vertex deuterated the drug, as the patent will run out in a few years, so that it stays around longer. They paid $400 million for the deuterated form, and the patent is coming out, and there'll be a new drug soon with a new patent life that you take once a day. So a longer half-life. Another sort of lesson to think about in the small molecule world. A kind of a strange but true. And I'm sorry, the second question?
Being an ion channel opener, do you anticipate sensitization?
Well, part of the question is off-target effects, right? That sensitization so much. It binds directly to CFTR, and there haven't been problems like that. And a lot of channels have been looked at. Whether you'd get tachyphylaxis, whether with time something would change. The follow-up on that particular drug, the potentiator, the one that's approved and most useful, is about 12 years now, and it's still working well. But that doesn't mean in 22 years something won't happen. But so far it hasn't been a problem, which is kind of comforting, for all of us. I think that you can give drugs for a long time and maybe you'll be okay. Certainly, developing resistance is or sensitization. Both of those are things.
With Dr. Sorscher talks ends the 2024 Bellucci Symposium. So we want to thank everyone for being here, especially the speakers for their talk. And I hope everyone goes back home with some new ideas and that everyone enjoy this symposium. And a big applause to all the speakers here, please. And to the audience too, the people in Zoom and the people here that came from different parts of the US, especially to be part of the Bellucci Symposium. I hope you enjoy, and we'll see you maybe next year for the 2025.